Hey team, Dr. G.A.M.E.D here, and today I'm going to continue to head through the AstraZeneca paper that I've been heading through. There was a brief interruption there while I had to introduce 3 different techniques that they're about to cover in this paper. They're all very similar techniques, just different versions of ELISAs, very cool, and now I'm going to cover them in this paper. So let's jump into it. Oh, let me get my laser pointer out. There we go. Okay, so this section is all about antibodies. Now remember, there's lots of different kinds of antibodies in your body. There are 5, but then there are subtypes that we're going to just allude to a little bit later. So here they are, and just really briefly, I'm just going to summarize them. So IgG is the antibody. It's the #1 antibody in your blood. It does most of the antibody work. Once it's fully matured through the antibody binding affinity processes of your body, IgG is a very high affinity antibody that floats around your blood. Now, IgE is called the allergy antibody, and it's really associated with allergies. It binds to antigens, and then the IgG binds to the Fc receptor on mast cells and basophils, which causes histamine release. And that's why we associate it with allergies, and because histamine is that allergic response going on, the massive swelling response, and so on and so forth. And it's really important in the Th2 response, because it helps us with the basophilic and mast cell responses needed to eliminate parasites from our gut, for example. Then there's IgD. This is most famous for being on the surface of B cells as the B cell receptor. So it's a membrane-bound antibody, often in B cells, and it acts as the B cell receptor. There's IgA. This is the mucus antibody. I remember it by the IgA antibody often goes out to your anus, and that's because it is often secreted in the gut to fight gut parasites. But it's also produced in other mucus places. It can go into your lungs, for example, to help defend against pathogens that are in your lungs. It can also come out of breast milk, for example. And then there's IgM. Now, IgM is a lot like IgG, except it's first. It's early. It's our early antibody, and it floats around the blood, and it does that agglutination, the precipitation, all that kind of stuff. It's especially good at agglutination and precipitation because of all of those binding domains, all attached to the one molecule. Right, now let's head on to the graphs. So here we have the graphs here, and again, in blue we've got the meningitis vaccine, and in red we've got the AstraZeneca vaccine. And here they have done the antibody ELISAs, that I talked to you about in the previous videos, to measure the levels of antibodies that would bind to spike proteins at different time points. Down here we've got the N, so there's 44 in each group, 44 little dots all in each group there. I might have jittered this graph, which means that the dots don't overlap, so you can see them a bit better, but hey, that's me. Okay, so we've got day 0. We can see that there's nothing going on. There's no significant difference there in these antibody levels. Now, here we've got IgM, which is the early antibody, and IgA, which is the secreted antibody. And we're looking at those levels. So red is vaccinated people for SARS-CoV-2. Blue is vaccinated for meningitis. So you really wouldn't expect any anti-spike proteins in this group. And indeed, if we look at blue, it stays around about the detection limit the whole way through the assay. It doesn't go anywhere. But in red, we see at day 7 some IgM, at day 14 lots of IgM, at day 28 lots of IgM, and at day 56 a little bit less. Now in the green here, this is really, really cool. They've got convalescent plasma. So this is plasma taken from people who are in the hospital and have tested positive for SARS-CoV-2. And this included health workers as well as patients. So these people have had SARS-CoV-2. And because right now it's looking like there is either incredibly low or no probability of getting SARS-CoV-2 within a year, because we've had the pandemic for about a year now. And we've seen few, if any, we're not sure, but it seems like there are very rare cases of getting it again. Once you get it once, it looks like you've done. So if we can get similar antibody responses to people that have had SARS-CoV-2 with our vaccine, we can say that this is a pretty good proxy and that our vaccine should work, because if we're getting the same levels of antibodies as having SARS-CoV-2 with our vaccine, that means we've replicated the disease. We've made the immune system think that it truly had the disease, and we got a fully fledged response there. Now, so IgM, I would say those levels are pretty similar, so they've done a pretty good job there, and IgA is the same. It's a little bit higher here, but remember, this is day 56. Convalescent means that they are probably currently tested positive, so you would expect more antibodies there. But that's pretty good in terms of IgM and IgA. Million miles an hour. So this is IGG Avidity. Now, avidity is an interesting word. What it means is the strength of bond between IgG and the antigen, the spike protein. So how strong is that bond between the IgG and the spike protein? Another way of saying that is, yes, we're producing antibodies, but are those antibodies good antibodies? Which is a great question. Oh, hang on, let me just nip back. So how did they know it was IgA or IgM? Remember, they have different tails, right? You know, that non-variable region of the antibody is different. So you can take that non-variable region of the antibody and inject human IgA into a mouse, and it will produce an antibody for human IgA. Or you could inject IgM into a mouse, and that mouse will produce an antibody for IgM. You can then use that mouse antibody as your detection antibody. So that's how we can tell which antibody subtype we've got in our plasma. Now let me jump back to this. We're measuring the strength of the bond between these antibodies. Have we produced good antibodies? Now remember, this is using the chaotrope, the intense chaotrope ELISA, where you basically put in a molecule that disrupts the interaction and see how resistant it is to that disruption as you increase the concentration of the chaotrope, which is a chaos-inducing agent. So as we add a chaos-inducing agent to it, can we break apart that bond? At what concentration does that happen? And this is a relative measure. Remember, it's not an absolute measure. And this is the chaotrope they used here, sodium thiocyanate. So this is the chaos agent here. And what we can see here is that we can see that the antibodies are getting better over time. And in future videos, you will learn why that's happening. But you can see at day 56, they've got some really good antibodies, and it's as good as people who have had SARS-CoV-2, right? So we can say that we're producing good quality antibodies with really strong binding affinities, which is really good results so far. Now, I haven't covered this, and I'll be covering it in a future video, but there are IgG subtypes. So within the G.A.M.E.D, IgG, IgA, IgM, IgE, within the G.A.M.E.D antibodies, IgG, G.A.M.E.D, I should say. Within the G.A.M.E.D antibodies, under G, there are further subtypes. Now, these subtypes do different things. They have different abilities to activate Fc receptors. They have different abilities at binding to proteins versus carbohydrates. But a good thing to know is that IG1 is a good one. IG1 is the high affinity protein antibody. It's a good one that we want, and here is our IG1 antibodies. I will say that those 95% confidence intervals are close to going over the bottom limit of detection. So I'm not sure if this is a significant increase. And also, I wonder, where is the anti-SARS-CoV-2 spike IgG subtypes for the meningitis group? How come we're not showing that? Where has that group disappeared to? So it would be good to have your blue dots there as a comparison. I hope that the blue dots were just non-detectable. Right. Oh, I froze there for a second. So this is the ELISA spot assay. Now remember, this is in a previous video. I covered what an ELISA spot is, but basically it's looking at T cells' ability to be presented antigens from monocytes and presented the spike protein antigens from monocytes and produce interferon gamma. So it's looking at, are there a lot of T cells that can recognize the spike protein floating around these people's blood? So they use PBMCs there. peripheral blood mononuclear cells, which includes monocytes and T cells. So you take those cells, you give them a spike protein, and then you count the number of cells that respond by producing interferon. And that's where that a live spot precipitate colorimetric assay comes into it. And what we can see is they've used the spike 1 domain and the spike 2 domain separately to see if there's a difference in what's going on. And what we can see is that the vaccine, so day 0 is pre-vaccine, day 14 is 14 days post-vaccine, S1 is spike 1, S2 is spike 2. And we can see that we get a lot of interferon cells responding. We get a lot of T helper cells responding. They're recognizing that spike antigen and they're releasing interferon. So this is cell number on the y-axis. And you can kind of think of it as percentage. So we're getting close to 0.1% of cells were responding, which shows you, you've still got a lot of other cells floating around your body, and 0.1% of those cells produce interferon, which suggests that you are bumping up your T helper cell numbers, and those T helper cells that can specifically recognize that spike protein. Then they also looked in these cells what at day 14, how much of each cytokine they would produce. This is pretty similar to that previous one. And they compared CD4 positive cells to CD8 positive cells. I haven't really covered this, but CD8 positive cells also produce cytokines to facilitate the immune response, as well as doing their cytotoxic function here. And we can see here the IL-5, which is a Th2 response, is very low. Again, this is a log scale here. And interferon and IL-2 are very high, and those are Th1 responses going on here. So it's very similar to the previous stuff here. And we've also got a Th1 response, well, a cytotoxic T cell interferon response going on here. Interferon alpha, TNF alpha is also a Th1 cytokine. So to summarize, have they got it in the right spot? Well, on the most part, it looks like maybe, right? Because they've got a reasonable amount of IgG, they've got a reasonable amount of IgA and IgM, so they are getting antibody production, but they are also getting a heck ton of T helper cell and cytotoxic T cell responses going on there, and they've got none of the Th2 cytokine levels here. And so did they quite get that balance right? Did they get the right balance between a Th1 and a Th2? And it's hard to know, and it's impossible to know from this data, is that the perfect response for that vaccine, for that virus? But to me, it looks like it's a much more of a Th1 response, right? You didn't get a huge amount of B cell proliferation in the flow cytometry graph. You didn't get a lot of IL-4 production from your T helper cells. So I'm starting to wonder, did they get the right balance right? Or is this too much of a cytotoxic T cell response and not enough of a Th2 response? And here is where some of the answer might lie. So there was an error in the stage 3 clinical trial. So they're doing a stage 3 clinical trial. Their goal is to reach 30,000 patients. They do actually continue it on and get to 40,000, I think, by the end of it. But they had a manufacturing error. You wouldn't believe it. They had a manufacturing error, which meant that the vaccine dose in solution was half the concentration it should have been. So about 3,000 patients in California got half the dose they were supposed to get, which is crazy. And it seems like a mistake, and it is a mistake, 100%. That was a mistake. And there was a manufacturing error, and it clouded the results, and it produced half the concentration that they wanted, right? Which is a bit of an issue, but you wouldn't believe what happened. Here's what happened. The people that got the full dose, it was around 62% effective. The people that got the half dose, it was around 90% effective. So this manufacturing error resulted in a key discovery, and it meant that, holy moly, we've got a way more effective vaccine than we thought. And so then it basically went out in the UK and Europe and a few other countries. It didn't go out in America initially. at the half dose. So they now said, okay, let's do the half dose. And they actually continued the phase 3 clinical trial using the half dose to make double, triple sure. And once they continued the trial and collected more, this is why it went over the original planned 30,000 patients, they actually bumped that number right up to 95% effective. What do we mean by effective? Preventing hospitalizations. They didn't go back. In this case, they just wanted to see, did we reduce the hospitalizations of SARS-CoV-2, and they found it 95% effective with that half dose. Here's what's super interesting about that, right? We have known for quite a while that the dose of the PAMP, the dose of the pathogen-associated molecular pattern, can influence the cytokines that are being released, which can influence the TH response. So this is a preclinical study in mice, and they're giving a vaccine. They're giving a vaccine with an antigen, and they're trying different adjuvant concentrations. So remember, that's that innate immune irritating thing that we put in vaccines to induce that innate response. Here's the no adjuvant result. And here we've got cytokines IL-4, IL-10, and interferon. And without an adjuvant, we get a very low response, right? There's not a lot of cytokine being released without an adjuvant. If we put in a high adjuvant concentration, so we get a high innate immune response in this case, we see the interferon in the dotted line here is through the roof, 250 picograms per mil. We've got loads of interferon. We've got a little bit of a Treg response. Remember, I always said that whenever there's one response, there's also the restraining response going on. So IL-10 is a regulatory T cell response, and we can see we've got a bit of a Treg response there. But we have no IL-4 here. No IL-4. IL-4 is in black. So with the high dose of adjuvant, we're getting no IL-4. With the low dose of adjuvant, we're getting IL-4. So we have known for a while that getting that dose right can dictate whether you get a purely Th1 response or a mixed response when it comes to vaccines. And so this could be, I'm not saying this is, but this could be the explanation as to why the lower dose vaccine was way more effective. It got that mixed TH response going on. So perhaps these guys would have a better production of antibodies. If you think back to that World Health Organization table that I showed in a previous video, the best predictor of most vaccine efficacy is not the presence of cytotoxic T cells, it's the presence of antibodies floating around the blood. So perhaps when they dropped that dose, they got more of a Th2 response going on, which meant that they got a greater production of antibodies in the blood. And so those people were more protected from SARS-CoV-2. They get more of a B cell response. They get those memory B cells being produced, and they get that greater level of background level of antibody going on. So what's interesting is in that phase 1 clinical trial, I think we can see hints, phase 1, phase 2 clinical trial, and then we can see hints of what was going on and what we now know. We saw the IL-8 release. Perhaps that's what's causing the clots. We saw that there wasn't any IL-4 production going on. Perhaps that's why dropping the dose really helped and getting more of that mixed Th1, Th2 response. So I think by dropping the dose, they shifted where it was in that spectrum, and they got to that nice little comfort zone between Th1 and Th2. This is just a hypothesis though, but I think it's really interesting, and I think it's probably what's going on, which I think is fascinating. It's a great example of why in most cases with vaccines, we want a mixed Th1, Th2 response going on. And I think it's a fantastic little case study to look at. Thanks everyone. That was the journey through the AstraZeneca vaccine. Feel free to comment below any questions and I will hit you up with some answers. Thanks.